Good evening, and thank you for joining me for our discussion on the physiology of skeletal muscle contraction. My name is Lindsay Sammons. I'm in the Biology 18 Physiology course at Merced College in summer of 2020. So before we dive into the physiology of skeletal muscle contraction, it's important to understand a little bit about the tissue itself. So skeletal muscle is one of three muscle tissue types within the human body, and it's the only voluntary muscle tissue type. So characteristically, it has these alternating light and dark bands all along the muscle fiber, and those are called striations. These skeletal muscle cells also have multiple nuclei, and they're cylindrical shaped. There's a few characteristics very important for skeletal muscles to be able to perform physiologically the way they do and by the mechanisms that they do. They have electrical excitability, which means they can respond to an action potential. They have contractility, extensibility, and elasticity. So let's look at the macroscopic anatomy of a skeletal muscle cell, or a skeletal muscle. So we're going to start with the short head of the biceps brachii and make a transverse cut and look down on it. So we're looking at our muscle belly here, and it's covered in an epimysium, which is a connective tissue covering. If we look deep to the epimysium, we find smaller subunits, each called fascicles. These fascicles are actually bundles of individual muscle fibers or cells, and they're covered in a connective tissue covering called a perimysium. If we look inside these fascicles, we're going to find even further smaller subunits. Those subunits are actually individual muscle fibers or muscle cells. So these are covered in a connective tissue covering called an endomysium. So we're going to take one of those muscle fibers and we're going to blow it up and look at it. So now we're looking at the microscopic characteristics of the muscle fibers. So we're still looking at our endomysium here, which is our connective tissue covering. So if we look deep to the endomysium, we're going to find the actual start of our muscle cell. So we're going to find our plasma membrane, which is called a sarcolemma. If we look deep to the sarcolemma, we're going to find the sarcoplasm or cytoplasm of the muscle fiber cell. So within the sarcoplasm, again, we're going to see even smaller subunits. Each one of these subunits is called a myofibril. Inside the myofibril is where contraction actually occurs. So the myofibrils contain functional units called sarcomeres, and each one of these myofibrils is wrapped in the sarcoplasmic reticulum. So we're going to take this muscle fiber and we're going to look at it lengthwise. So we're looking at our skeletal muscle fiber here on the left at the top, and we have individual myofibrils. So if we look on the myofibrils in the picture below, then we can see that they're covered in that sarcoplasmic reticulum. The function of the sarcoplasmic reticulum is to store and release calcium, which is necessary for skeletal muscles to contract. We can also see these yellow lines transversing this muscle fiber. Those are called transverse tubules, and they penetrate the muscle fibers at right angles to the myofibrils. So if we cut away some of that sarcoplasmic reticulum, we can actually start to see some of the functional units below, the actual sarcomeres. So the sarcomere runs from one Z disc to the next Z disc, and they are separated by an M line, which is central to the two. So we have these alternating I bands and A bands, which are actually the striations that we talked about in the beginning, the alternating light and dark regions of the muscle fiber that runs the length of the entire cell. Those are made out of proteins. We call those myofilaments. So we have two types of filaments in skeletal muscle cells. We have thick and thin filaments. The thick filaments are primarily composed of the protein myosin, and the thin filaments are primarily composed of the protein actin. The myosin is anchored to the M line. The actin filaments are anchored to the Z discs. So this is where contraction is actually going to occur. But in order for this to happen, we have to have stimulation from the nervous system. These are voluntary muscles, so they need an action potential to be propagated along a somatic motor neuron to the space where the nervous system meets the skeletal muscle fiber, which is at the neuromuscular junction. So the neural end of this junction is the presynaptic end bulb, the swelling at the terminal end of the axon. That bulb is full of synaptic vesicles, which contain neurotransmitter molecules. In the case of skeletal muscle contractions, those neurotransmitter molecules are going to be acetylcholine. So when the action potential arrives, it arrives in the presynaptic end bulb, which stimulates those synaptic vesicles to release the acetylcholine. So the presynaptic end bulb doesn't actually touch the skeletal muscle fiber. There's a space between the two, and that's called the synaptic cleft. So the acetylcholine is going to diffuse into the synaptic cleft and cross to the motor end plate, which is the skeletal muscle end of the neuromuscular junction. So embedded in that motor end plate are receptors specific for the acetylcholine. So as the acetylcholine binds to the receptors, it's going to cause the voltage-gated sodium ion channels to open, allowing sodium to diffuse into the cell. So the sodium gates opening causes a sequential opening of further sodium gates that propagates the action potential along the entire sarcolemma or plasma membrane of the cell. Now remember we said those transverse tubules, which were invaginations of the sarcolemma. So those are part of the sarcolemma. So that action potential is also going to be propagated along the transverse tubules. So if we look at our actin and myosin, we see in this top picture on the right that we have some regulatory proteins embedded in that actin. We have troponin, which forms a troponin complex with the actin, and tropomyosin, which actually binds the myosin binding site on the actin filament. So when this action potential is propagated along the sarcolemma and through the transverse tubules, it's going to stimulate the terminal cisterns, or the dilated end sacs of the sarcoplasmic reticulum that surrounds every myofibril, to release their stored calcium. That calcium is going to diffuse into the cell, into the sarcomere, where it will bind with our troponin complex. This causes the tropomyosin that's blocking that myosin channel to shift, allowing the myosin to actually bind to its binding site on the actin. Now, in order for this to occur, ATP will have already been present in the unit. So ATP will have been bound to one of the myosin heads. The ATP is going to undergo a hydrolysis in order to create ADP and inorganic phosphate. The inorganic phosphate will be released, which allows the myosin to bind to the newly exposed binding site on the actin filament. This initiates the sliding filament mechanism. So the myosin head is bound to a myosin site on the actin filament, and this initiates a power stroke. So the myosin head is going to shift to a high energy state, which pulls the actin molecule towards the M line. Now remember, the actin is anchored to the Z disc. So the Z discs moving towards the M line causes the sarcomeres to shorten. So since the muscle fiber is a sequence of repeated sarcomeres all along the muscle cells, the sarcomere shortening causes all the sarcomeres to shorten, which causes the skeletal muscle to contract and shorten as a whole. So this movement of the myosin head from a low energy state to a high energy state is called a power stroke. That power stroke in the process causes that ADP that's still attached to the myosin head to be released. And this forces the myosin back to its original position, which is a low energy state. Once it's back in its low energy state, this allows a new ATP to come along, rebind to that myosin head, and the process repeats. This is a physiological process of the contraction of skeletal muscle. Here are the references. Thank you.